So today we will learn the chapter 8. The title is Protein Synthesis, Processing and Turnover. Okay, today we will cover 4 subjects starting from the transcription, translation, and antibiotics provides insight into protein synthesis. Finally, we will cover protein sorting or targeting. So in this slide, I will explain the first subject, transcription. Okay, so this is the DNA sequence. Okay, for example, this is a transcribed region, and this is promote region. Okay, for transcription initiation, so we need the Tata box. Okay, so usually the transcription factor 2D, we call it as a TF2D. Tata box is the TF2D is binding site. So usually TF2D is bind here, and Tata box is composed of the 35 box, minus 35 box, and also minus 10 box. So minus 35 box has a consensus sequence which has the TTGACA, and also minus 10 box also has a consensus sequence which is composed of the TAT, So between the minus 35 box and minus 10 box, there are optimal interbase distance is there. So usually the 17 plus minus 1 base pair region is located here. So this is the Tata box. Tata box plays important role in the transcription initiation. So we will see some details function in the later. Okay, so in this slide, I will explain the eukaryotic RNA polymerase. So table 8.3 summarizes the role of eukaryotic RNA polymerase and their location. So there are several kinds of RNA polymerases there. First one is the RNA polymerase 1 and RNA polymerase 2 and RNA polymerase 3 and the mitochondrial RNA polymerase and chloroplast RNA polymerase. So in this chapter, we will focus on RNA polymerase 1 and RNA polymerase 2. So let me start RNA polymerase 1. So RNA polymerase 1 is located within the nucleus. So this is cell. Cell has the nucleus. Okay, so RNA polymerase 1 is located within nucleus. However, especially the RNA polymerase 1 is located within nucleus, especially in the nucleolus. So nucleolus means in Korean we call it as in. So RNA polymerase 1 is located within the nucleolus. Okay. So the main function of RNA polymerase 1 is to transcribe the pre-rRNA except for the 5S rRNA. Okay. So we will see some detailed information in next slide. And also there is the RNA polymerase, okay, and also there is RNA polymerase 2. RNA polymerase 2 is located within the nucleus, not especially in the nucleolus. So RNA polymerase 2 is located within the nucleus. So their main function is to transcribe pre-mRNA, and sometimes transcribe some small nuclear RNAs. But mainly, RNA polymerase 2 is transcribed pre-mRNA. Okay. So in this slide, I will explain the RNA polymerase 1. The main function of RNA polymerase 1 transcribed 55S free rRNA, coding for ribosomal rRNA, especially the 18S and 5.8S and 28S rRNAs. So, okay, so 45S free rRNA is the tandemly repeated in the DNA. So this unit and this unit, this unit, this unit, this unit, maybe 1,000 times is repeated this unit. Okay. So at that, RNA polymerase 1 is transcribes each subunit. So they will make the 55S free rRNA. So that this 55S free rRNA is composed of the 18S rRNA and 5.8S rRNA and 28S rRNAs. After that, multi-step RNA processing happens. So finally, they will make the 18S rRNA and 5.8S rRNA and 20S rRNA is made is like this. Okay, especially in 18S rRNA. So, okay, so 18S rRNA is combined with 33 ribosomal proteins. So they combined with 33 ribosomal protein. After that, they will make the small poly as subunit. So they will make a small poly as subunit is like this. How about the 5.8 srRNA and 20 srRNA? So in this case is that 5S rRNA is added, and also additionally 49 ribosomal protein is added. So they will make the large 60S subunit is like this. Okay, so for example, 5S rRNA, especially in this case, RNA polymerase 3 is transcribes the 5S rRNAs. So however, the RNA polymerase 1 is transcribes 45S free rRNA coding for ribosomal rRNA, especially the 18S rRNA and 5.8S rRNA and 28S rRNAs. It's like this. So in this slide, I will explain the RNA polymerase 2. So main function of RNA polymerase 2 is transcribes genes coding for free mRNA and small nuclear RNAs. So this is the table 8.3 summarized the role of the RNA polymerase 2. So main function is the transcribes genes coding for pre-mRNA and some small nuclear RNA, but we will focus on the pre-mRNA synthesis. Okay, and also RNA polymerase 2 requires basal transcription factors. So table 8.4 summarized basal transcription factor required by eukaryotic RNA polymerase 2. So there are several factors starting from transcription factor D and A, B, E, F, H. So we will see some detailed function in the next slide. Okay. So in this slide, I will explain the pre-initiation complex. Okay, so for transcription initiation, pre-initiation complex should be made. Okay, pre-initiation complex is composed of transcription factor 2D and 2B, F, RNA polymerase 2, and E and H. So each has a specific function. Okay, let me start transcription factor 2D. So we call it as the Tata box binding protein. So this is 2D. So 2D can recognize the Tata box. After that, it can bind to the Tata box. Is like this. How about the 2B? So 2D is the recruit 2B again. So this is the Tata box transcription factor 2B. So the main function of 2B is that start site selection. So this is the initiation site. So this 2B is scan the region. After that, it will find the transcription initiation site here. Okay. How about the next? 2F is bind to the here. Okay, 2F is bind to the here. The function of the 2F transcription factor 2F, F is that target the RNA polymerase 2 in the promote region. So after the F is bind to here, it recruits the RNA polymerase 2 in the promote region. Okay, after that RNA polymerase 2 bind to the promote region. Okay, Tata box region and also the promoter region is like this. After that, it will recruit transcription factor 2E. So main function of 2E is that recruitment and the modulation of the H activity. The main function of 2E is recruit transcription factor 2H and also the modulate TF2H activity. Also, the main function of H is that it has the helicase activity. So it means that it will the main function is promoter melting. So it will unwind the DNA region. So it can after that it can initiate the transcription initiation like this. Okay. So each of the basic transcription factor has the specific function starting from the 2D, 2B, 2F, RNA polymerase 2, after that E and H is like this. Okay, so in this slide I will explain the TF2D structure, transcription factor 2D structure. So we call it as the Tata box binding protein, or in another words, TBP. Okay, so this is the TF2D, and this is DNA. So this is DNA structure, and this is the TF2D, or Tata box binding protein structure. Is like this. You see the structure. Structure looks like the saddle shaped structure. So this is a horse. So the above one is a saddle. Okay. Their structure looks like the saddle-shaped structure. Okay, they are one protein. So they are one protein. N terminal is starting from here, and C terminal is located here. Okay, but they look like that they have two subunits. So this is the left subunit, and this is the right subunit. Okay, so each subunit is composed of the 5 antifederal beta sheet. Okay, how about let's start from the left one. Okay, 1 antifederal. Okay, after 2 antifederal, 3 and 4, 5. Okay, so 5 after that they have the alpha helix. Okay, between the 1 and 2nd antifederal, there is another alpha helix is located here. Okay, so once again, in this slide, we cover transcriptional factor 2D structure. They show that the saddle-shaped structure. So they are one protein composed of starting from the N terminal to C terminal, but they looks like they have the two subunits. Which one is a left subunit and right subunit. So each subunit is composed of the 5 antifederal beta sheet and 2 alpha helices. The total 10 antifederal beta sheet and 4 alpha helices. Okay. So in this slide, I will explain the transcriptional factor 2A function and structure. So transcriptional factor 2A is not belong to the pre-initiation complex, but they have some special function. Their function is that stabilization of the Tata box binding protein or binding, TBP binding. So they have specific structural motif. So they have the zinc finger DNA binding motif, which is composed of the cysteine, and X means that any amino acid go to the here, and after that C is the cysteine, and X means that any amino acid go to here. So X12 means that about 12 amino acid come to here, and H means that histidine, and X means that any amino acid come to here, and H means that histidine. So this is the zinc-Fing DNA binding motif, and transcription factor 2A has this motif, position this motif, and this motif, and this motif, and this motif, and this motif is like this. Okay, so if you see the zinc-Fing DNA binding motif in the structure, for example, this is cysteine, so they have the sulfur and hydrogen, and sulfur, hydrogen. Okay, after that they also have histidine, so this is the histidine, so this is the nitrogen, and nitrogen, and in the center there is the zinc. Zinc cation is here. Okay, so between the zinc and histidine, nitrogen or H, so there is the hydrogen bonding is exists. Okay, so this is the zinc-Fing DNA binding motif, and transcriptional factor 2A has the several zinc-Fing DNA binding motif. Okay, so transcriptional factor 2A exactly fit to the major group of the DNA. So DNA is composed of the major group and the minor group, and transcriptional factor 2A exactly fit to the major group of the DNA. After that, it will stabilize the tartar box binding proteins binding. So this is we learned transcriptional factor 2A of function and their structure. Once again, they have the zinc-Fing DNA binding motif, which is mainly composed of the cysteine and histidine and histidine. It's okay. So in this slide, I will explain the second subject, translation. So translation means that conversion of genetic information into the protein. So this is recorded as the translation. So first of all, we have to know the genetic code. So genetic code means that rules to translate genetic information into proteins. And also we have to know the reading frame. So reading frame means that initial triplet of the nucleotides from which translation starts. Okay, let me see some example in the reading frame. Okay, so figure 8.32 shows that translation of synthetic polyribonucleotides. So AAG, 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 AAG. So this one is lead to the production of 3 different amino acids in cell-free translation system. The first case is like this. If the reading frame is like the AAG, 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 in this case is that it will encode the lysine. So it will make the lysine, 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 lysine. Second case is that reading frame is that AGA, 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 AGA. So in this case is the AGA is encodes the arginine. So it will make the arginine, 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 arginine. The final case is that the reading frame is GAA, 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 GAA. So in this case is GAA is encodes the glutamate. Okay, so it will make the polyglutamate is like this. Okay, so we have to know the reading frame. Once again, reading frame is initial triplet of the nucleotides from which translation starts. Okay, and also we have to know initiation and termination codon. The initiation codon is recorded as AUG. AUG encodes the AUG is the initiation codon. So AUG encodes the methionine. How about the termination codon? So in the termination codon, there is three different termination codon is available. First one is UAA, and the second one is UAG, and the third one is UGA. So in this case, for example, start codon, initiation codon is encodes the methionine. However, the termination codon, these three termination codon did not encode the amino acid. Okay, this one is just the stop. Okay. So in this slide, I'll explain another two special characteristics in genetic code. First one is the codon degeneracy. Second one is the genetic code is nearly universal. However, there is some exceptions. Okay, let me start the codon degeneracy. Codon degeneracy means that redundancy of the genetic code is exhibited as the multiplicity of three nucleotide combination that specifies an amino acid. Okay, let me show you one example. For example, in this case, UUA, UUG, CUU, CUC, and CUA, and CUG, these six different three nucleotide combination specify the leucine. So this is the one example of the codon degeneracy, redundancy of genetic code. Another example is the, for example, UCU, UCC, and UCA, UCG is also specify or encodes serine. So this is the another example of codon degeneracy. Okay, the second characteristic is genetic code is nearly universal. Okay, for example, so this genetic code is applied to all of the prokaryotic cell and nearly all of the protozoan or nearly all of the eukaryote. However, there is some exception. For example, this codon AGA and AGG is universally encodes arginine like this. However, in some animal mitochondria or in some protozoan, it can be used as a stop codon or the serine. So this is the exception. This is the one extremely exception. It's not nearly happen, but there is some exception. Okay, so for example, some animal mitochondria or in some the protozoan, so this genetic codon can be used as a stop codon or serine. So there is another exception is there. So we will see some details. Okay. So once again, to another characteristics in genetic coder, that is the codon degeneracy, and also genetic code is nearly universal. But however, there is some exception. For example, in some mitochondria or some protozoan and some prokaryotic cells. Okay. So in this slide, I will explain the transfer RNAs. So we call it as tRNA. So this is the tRNA structure. So this is 5' and this is 3'. So from the 5' there is a D loop, and also there is the anticodon loop, and also T loop, and finally they have the amino acid attachment site. Okay, let me explain some important important characteristics in the tRNA. The first one is the anticodon. So this is anticodon. And second one is the tRNA has the amino acid attachment site at the 3' region. Okay, so the function of anticodon is that 3 nucleotide sequence complementary to that of the corresponding codon in the mRNA. Okay, so in this case, this is the mRNA starting from 5' is located here and 3' located here. The codon is that GAG. So codon is the GAG. So in this case, GAG is the it encodes the glutamate. So in this case, codon was a GAG. In the anticodon was the 3 nucleotide sequence complementary to that of the corresponding codon. So anticodon is a CUC. Okay, so this is the anticodon. CUC is anticodon. This is the complementary to that of the codon. And after that, the main function of the transfer RNA is tRNA forms base pairs with the corresponding codon in the mRNA and ensuring that appropriate amino acid is inserted into the amino acid attachment site. So in this case, glutamate can be attached to the amino acid attachment site. Okay, so this is the main function of tRNA. So once again, the most important characteristics in the tRNA is the tRNA has the anticodon, and also tRNA has the amino acid attachment site at 3' region. So anticodon means that 3 nucleotide sequence complementary to that of corresponding codon, and also amino acid attachment site is that the tRNA forms base pair with the corresponding codon in the mRNA, after that ensuring that appropriate amino acid can be inserted into the amino acid attachment site. In this case, is glutamate, codon was GAG, so in this case, glutamate can be attached to here. So in this slide, I will explain the amino acid tRNA thindase. So amino acid tRNA thindase is divided by two enzymes, class 1 and class 2. So let me start the class 1 amino acid tRNA thindase. So this is class 1. For example, this is the amino acid class 1 amino acid tRNA thindase. So this is tRNA. So class 1 is bind to the back of the tRNA. It's like this. So okay, the main function of class 1 amino acid tRNA thindase is attaches the amino acids to tRNA, especially the cysteine, glutamic acid, and glutamine, and finally the valine. Okay, how about the class 2? So this is the class 2 amino acid tRNA thindase. So class 2 amino acid tRNA thindase attaches the amino acid to tRNA, especially the alanine, asparagine, aspartic acid. Finally, there is a threonine. Okay, and also there is some specific difference between the class 1 and class 2. Okay, so this is the tRNA structure. I magnified the tRNA. So this is 5' and 3' is located here. Okay, 76 nucleotide is adenosine. So in the tRNA, 76 final nucleotide is adenosine. Okay. So, but class 1, so this is the adenosine is located here. Okay, they have in 2' location they have OH, and 3' location, they also have OH. So class 1 amino acid tRNA thinase attach amino acid to 2' OH of adenosine in the tRNA, especially here. Here, so this is the 76th nucleotide is adenosine, especially it's a class 1 amino acid tRNA thinase attached amino acid to 2' OH. How about class 2? Class 2 attached to amino acid to 3' OH. So this is the main difference. Okay, so let me start some of the specific details in the amino acid attachment procedure. Okay, so this is the amino acid and this is the ATP. So amino acid and ATP is combined. They will make the amino acid intermediate. So this is amino acid. They have a carboxyl group here. So for example, class 1 is the attached, this OH is attacked here, or class 2 is this one OH is attached here. In class 1, this OH is attacked here. So they will make the tRNA and the amino acid is like this. So 76 nucleotide is adenosine, especially in the class 2 amino acid tRNA thinase attached amino acid in 3' location, and class 1 amino acid tRNA thinase attached amino acid in the 2' OH. Okay, so this is the amino acid tRNA thinase. So in this slide, I will explain the ribosome. So ribosome plays a crucial role in the translation, especially in the initiation, elongation, and termination. So we will see some detailed mechanism in the next slide. Okay, and also ribosome consists of the two major components, the large subunit and small subunit, and also each subunit consists of the ribosomal RNA and the proteins. In previous slide, we learned that we focused on the ribosome, especially focused on the mammalian ribosome, which is recorded as ADS. So ADS is composed of the 60S large subunit and 40S the small subunit. How about in bacteria? In bacteria, we call it as 70S. So 70S ribosome is composed of the large subunit and small subunit. Okay. For example, in the large subunit, 23S rRNA and also 5S rRNA plus 31 ribosomal protein is combined together. After that, they will make the 50S subunit. How about the small subunit? So in small subunit, the 16S rRNA plus 21 protein is ribosomal protein is bind there. So they will make the 30S small subunit. So we will see some detailed function in the next slide. Okay. So in this slide, I will explain the translation initiation step at prokaryote. In the following slide, I will explain translation elongation and translation termination step at prokaryote. Okay, table 8.7 summarizes the soluble protein factors involved in E. coli protein synthesis. Okay, especially at initiation, IF1, initiation factor 1 and factor 2, 3 is involved in this step. Okay, so let me start IF1. So this is the IF1. So main function of IF1 is assist in IF3 binding to 30S ribosomal subunit. Okay, how about IF3? So this is a very important function. Okay, IF3 is the dissociate 30 as subunit from inactive ribosome. So this is inactive ribosome. So IF3 dissociate 30 as subunit from inactive ribosome and promotes the binding of the 30S subunit to mRNA. So IF3 is the promotes the binding of the 30S ribosomal subunit, so this subunit to the mRNA. Okay, so especially 30S subunit with IF1 and IF3 is attached to 5' end of the mRNA. After this subunit, this complex is moves along the mRNA until it finds a start codon. So in this figure, you will see that this complex is find a start codon, AUG start codon. Okay, so this is the first step in the translation initiation. Okay, so after that, IF2 bind to GTP and also bind to initiated tRNA. So initiated tRNA means that tRNAs carrying N-formyl methionine. So this is methionine and this is nitrogen. So in nitrogen location, the aldehyde is bind to in the nitrogen location. So in this case, we call it as the N-formyl. So N-formyl methionine. So this is the N-formyl methionine complex in tRNA. So this is, we call it as initiate tRNA. Okay. So once again, IF2 bind to the GTP and also IF2 bind to the initiate tRNA. Okay. So after that, initiated tRNA is bind to the start codon, the AUG start codon. So after that, so hydrolysis of the GTP in the IF2. So IF2 has the GTP, so but hydrolysis of the GTP in IF2 released IF1, IF2, IF3. So released IFs. Okay. After that, 50S subunit, the largest subunit is bind to the 30S subunit. So this is the translation initiation step. Okay. So in this slide, I will explain the translation elongation step at prokaryote. So we can divide the translation elongation step at 3 steps. So first one is codon recognition, second one is peptide bond formation, and third one is translocation. The first one is we call it as codon recognition. For example, like here, new amino acid tRNA. So that is complementary to next codon is bind to the A site. So this is codon recognition. Okay, the second procedure is we call it as peptide bond formation. So this is the initiate tRNA which has the methionine. So methionine moves from the P site to A site to forms polypeptide chain linked by the peptide bonds. So this is, we call it as a peptide bond formation procedure. So if you see some detailed information, you can see that you can hear. Okay, so this is methionine tRNA. Okay, they have the tRNA and connected by carboxyl group and amine group, and this is R group. Okay, so in this case, okay, actually this is methionine tRNA which has the carboxyl group. Actually the amino acid tRNA, for example, they have the amino group. So this amino group attack the carboxyl group. After that, peptide bond is making like this between the carbon and nitrogen. Peptide bond is making is like this. Okay, after that, the methionine tRNA will become the uncharged tRNA is like this. So uncharged tRNA is like this. Okay, so this is the peptide bond formation. So once again, methionine moves from P site to A site. So if you see some detailed information, actually the nitrogen is attack the carboxyl group. After that, peptide bond is making is like this. Okay, so polypeptide chain is making is formed. It's like okay, and also linked by the peptide bond. Okay, so after that, ribosome translocate to the next codon. Okay, so tRNA in P site after that leaves the ribosome, and also tRNA with the growing polypeptides lies at P site. So after that, as A site is open, so A site is open for the binding of next amino acid tRNA. So this step we call it as the translocation step. So translation elongation step, there is a three procedure. First one is the codon recognition. Second one is a peptide bond formation. Third one is the translocation. Okay, so this is we call it as trans elongation. After that, this step is repeated. Okay. Okay, so in this slide, I will explain the translation termination step at prokaryote. Okay, during the translation termination step, first of all, the stop codon UAG or UGA or UAA, stop codons enters A site. After that, release factor 1 or release factor 2 is recognized stop codon. For example, this is table 8.7 shows that soluble protein factors involved in E. coli protein synthesis, especially in termination. Release factor 1 is recognized UAA or UAG stop codon. How about release factor 2? So release factor 2 is recognized UAA or UGA stop codon. So release factor 1 or 2 is recognized stop codon. After that, tRNA with polypeptides becomes hydrolyzed. So water is added here, and this bond is hydrolyzed. After that, it releasing the polypeptides into the cytoplasm. After finally, the small and the large subunit of the ribosome and also tRNA and the release factors dissociate from the mRNA. So this is the translation termination step at prokaryote. Okay. So in this slide, I will explain the third subject. Antibiotics provide insight into protein synthesis. Okay, so in previous slide, we learned that amino acid tRNA synthase. So amino acid tRNA synthase is make the amino acid tRNA. So this is amino acid tRNA structure. So 5' starting from 5' and 3' is like this. So this is puromycin. So puromycin has a structure similar to the 3' ends of amino acid tRNA. So this is 3' ends of amino acid tRNA. So we can magnify the amino acid tRNA. So the puromycin has the structure similar to 3' ends of amino acid tRNA. So nearly similar except for this region and except for this and except for this region. So it means that puromycin can enter the A site and after that block the peptide bond formation. So puromycin enters at A site and blocks the peptide bond formation. So it means that it will cause premature polypeptide chain release. So it will induce the premature polypeptide chain can be released like this. Okay, so this is one example of antibiotics that inhibit protein synthesis. So in this slide, I will explain another example, antibiotics example, to provide insight into protein synthesis. Okay, before I go further, I will explain the one specific mechanism, catalytic mechanism within the large subunit of the ribosome. Okay, so large subunit of a ribosome has the adenine monophosphate group. Okay, so this is adenine monophosphate, which is composed of the ribose and phosphate and also adenine. So especially in adenine, in this blue nitrogen, we call it as N1 nitrogen, and the red nitrogen, we call it as N3 nitrogen. And also especially in the large subunit, there is P site is here. P site contains the tRNA with polypeptides. How about A site? There is the amino acid tRNA is located here. Okay. First of all, in the large subunit, the N3 of the adenine monophosphate receives the proton, the H proton from the NH2 group of amino acid tRNA. After that, nitrogen in the amino acid tRNA is attacked the carbonyl group of tRNA with polypeptides. Except that they will make this tetrahedral, 1-hedral, 2-hedral, 3-hedral, 4-hedral, tetrahedral intermediate. So this is the intermediate step. Okay. So after that, so this is the tetrahedral intermediate, and this is the oxygen with 3 pairs of electrons. So in this location, resonance happens. So the 1 pairs of electron attack this carbon. Okay. After that, the carbon is attacked the oxygen, and finally the tRNA with the oxygen is attacked the hydrogen. Okay, hydrogen in the NH group. Okay, so this oxygen receives the proton from this NH group. So after that, this tRNA will become the tRNA and OH. After that, so this oxygen is resonance happens like this. So this oxygen will become the carbonyl group is like this. After that, they will make the peptide bonds with peptide bond is like this. So this the tRNA will be located in the A site. So it will synthesize the tRNA with growing polypeptides. So this is the catalytic mechanism within large subunit of the ribosome. Okay. So in this slide, I will explain another antibiotics. We call it as a Yarus inhibitor that inhibit protein synthesis. The full name of Yarus inhibitor is CCDA phosphate puromycin. So this is puromycin structure is like this. So CCDA phosphate puromycin is here, and this is CCDA structure, and this is phosphate is here. So CCDA phosphate puromycin has a similar structure to tetrahedral intermediate. For example, especially in this region, so in this region is nearly similar to the tetrahedral intermediate. So okay, so this is tetrahedral intermediate, and after resonance happens, they will make this kind of structure. Okay, so CCDA phosphate puromycin has a similar structure, similar to the tetrahedral intermediate. So it means that Yaros inhibitor function as a comparative inhibitor of tetrahedral intermediate. So it means that Yaros inhibitor inhibits peptide bond formation. So for example, peptide bond formation should be happens in tetrahedral intermediate. However, Yaros inhibitor function as a comparative inhibitor of tetrahedral intermediate. So it means that finally this Yaros inhibit the peptide bond formation in the tetrahedral intermediate. Okay. This slide I will explain the fourth subject, protein sorting or targeting. So SRP mediated pathway plays an important role in protein sorting or targeting. Especially SRP means that a signal recognition particle SRP is recognized the signal peptide. So figure 8.72 shows one example. So N-terminal sequence of the secretory proteins. Okay, so the signal peptides for secretory protein this is the signal peptide in front of the N-terminal region. Okay, so signal peptides for secretion is composed of the basic residue and also composed of the hydrophobic core. At that signal peptidase cleaves this site. Okay. So nowadays the Celltrion and at Celltrion or Samsung Biologics and Samsung Bioepis, whenever they make the therapeutic antibody, so they always add the signal peptide for secretion. Okay. Nowadays, their main focus is they want to find the efficient signal peptide for secretion, but they cannot find the breakthroughs. But who is studying the signal peptide for efficient sorting, efficient secreting? Who is studying? It's me. I'm studying the signal peptide for secretion. So only I can find the solution. Okay. So this is the signal peptide. Okay. So usually the SRP recognize the signal peptides. So at that SRP translocate the proteins to the designated location from ER. So this is a rough ER and this is smooth ER. So SRP translocate protein to the designated location from the ER to Golgi ER to from ER to Golgi to in this case is their signal peptides for secretion. So in this case is SRP translocated protein to the designated location means that extracellular spaces. Okay, so this is the SRP mediated pathway. So in this slide, I will explain the structure and organization of the SRP. So SRP is composed of the N domain and G domain and M domain. Okay, so N domain is located in N terminal, and G domain is located in the middle. This is the largest domain, and the M domain is located in C terminal. Especially the M domain has the long hydrophobic groups. So we magnified this region here. So they have the long hydrophobic groups like this. Actually, this site signal peptide can bind to this region. So usually the many signal peptide has the hydrophobic sequence, and that signal peptide can easily bind to this long hydrophobic group is like this. can bind to the here. So this is the structure and organization of the SRP. So in this slide, I will explain the protein targeting to the mitochondria. Okay, for example, this protein has the N-terminal region. They have the basic residue. Okay, also there are several protein spaces located here. Okay, but we will focus on this protein, especially has the basic residue at N-terminal region. Okay, so signal peptide for targeting protein to mitochondria has the basic residue at the N-terminal region, including arginine or lysine. Okay, so this protein, okay, so the signal peptides can be recognized out membrane translocase system. We call it the TAM complex. So TAM complex is recognized the basic residue in the proteins. After that, the transport this protein is the inside. After that, so this is the mitochondria outer membrane, and this is the mitochondria inner membrane. Okay, after that, inner membrane translocase system TIM complex also recognize the signal peptides and transport proteins inside. After that, so this is the mitochondrial matrix. Okay, inside the matrix. So after that, signal peptides. So this is signal peptide. So this signal peptide is cleaved by MPP proteins. So this is the procedure how to target the protein to the mitochondria inside. Okay. Also, there are another protein is which has a target for the outer membrane, which has a target for inner membrane, but we will not cover in today's lecture. Okay. So in this slide, summarize the outer membrane translocase system TAM complex and also inner membrane translocase system TIM complex. Okay, table 8.9 summarized proteins identified as component of the outer membrane translocase system, which is starting from TAM5, TAM6, and finally there is TAM72. So each of the protein has some specific function is like this. And also there is the table 8.10 summarize proteins identified as component of the inner mitochondrial membrane translocase system. We call it the TIM complex. TIM complex. So TIM complex is start from the TIM8. And also there are several other protein is available, and they have some specific function is right down here. Okay. Once again, so mitochondria has the translocase system, outer membrane translocase system, and inner membrane translocase system, TAM and TIM. Okay. Both of them is recognize the signal peptides and the transport. First of all, the transport the protein is inside. After that, TIM complex is also recognize signal peptides, and finally it will transport the protein into the inside of the mitochondria. We call it as a matrix. After that, signal peptide is cleaved by MPP, another proteins. Okay. So in this slide, I will explain the protein targeting to nucleus. Okay, the signal peptide for targeting protein to nucleus, we call it as nuclear localization signal. We call it as NLS. So NLS has a preference for lysine or arginine. This is plus charge, and additionally they have the proline. Okay, so this is the table 8.11 summarized sequence identified to act as signal for localization to cell nucleus. For example, some protein has the NLS sequence, proline, lysine, 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 arginine, lysine, valine. For example, another protein also has NLS sequence, lysine, arginine, proline, arginine, proline. For example, if some protein has this NLS sequence, that protein is definitely transported to inside the nucleus. Okay, so in the protein targeting to the nucleus, so importin plays important role. For example, importin, nuclear import proteins, importin binds to the, so this is the cargo protein. They have the nuclear localization signal. So importin, so they have some space like this, okay? So we will see some structure in the next slide, okay? So I will cover the structure of importin in next slide, okay? So importin is bind to the NLS of cargo proteins, so they will make the importin cargo complex is like this. After that, this protein can be transported inside of the nucleus. Okay. So once again, importin binds to the NLS of cargo protein. After that, imports cargo protein is inside the nucleus. Okay, so this is the mechanism. So in next slide, we will see some the structural information of the importin. Okay, so this is the importin structure starting from N-terminal to C-terminal. If we see some detailed information of the importin, okay, so we will see the in here, okay, this is also importin. Okay, so each importin is composed of 19 subunits. Okay, each subunit is show that this kind of structure, helix, turn, helix. Okay, so this is the subunit, and also importin is composed of 19 subunits. So 19 subunits is like this. Okay, so this is the importin structure. So, okay, so each subunit has helix structure. Okay, like this. Okay, so this is the helix. Okay, each subunit has helix structure. So if we twist it again, so it will make the superhelical structure is like this. Each one has the helix, but again they made the superhelix structure like this. So we call it as this is a superhelix. Okay, for example, each subunit has a helical structure. Also, after they combine together, they will also make this kind of helix structure like this. Helix structure like this. They make this helix structure like this. Okay, so this is, we call it as a superhelix structure. Okay, so usually the cargo protein which has an NLS sequence is bind to here. Okay, so and also the NLS sequence in cargo protein is not removed even after cargo protein is transported inside the nucleus. In most of cases, signal peptide is removed after the protein is transported inside the mitochondria or inside the lysosome, inside another organelle. But especially in the nucleus, signal peptide is not removed. Why this kind of the phenomena is happen? Okay, so I will show you one case or the example, the reason. So this is our eukaryote. So for example, fungi, only fungi has the closed mitosis. So this is nuclear membrane. Okay, this is nucleus. Okay, so this is the only fungi has the closed mitosis. But except for fungi, nearly all of eukaryote, they have the open mitosis. For example, so this is the nucleus. Okay, so during the prophase, okay, during metaphase, the nuclear membrane is disappears. So this is recorded as the open mitosis. So, but is it during the telophase? So the nuclear membrane is appears like this. This appears like this. So if NLS sequence is removed, okay, if NLS sequence is removed, so usually the nuclear protein can be lost, okay, it's because the NLS is disappears. So because they are doing the open mitosis, so sometimes, so during the open mitosis, protein can be go to the cytoplasm, but if there is no NLS sequence again, the protein cannot be go inside of the nucleus. That's why NLS in cargo protein is not removed even after cargo protein is transported inside the nucleus. Okay? So today we learned chapter 8. So chapter 8 covers post-transcription and translation, and also antibiotics provides insight to protein synthesis. Finally, we learned protein sorting or targeting. Okay, so today we will finish class.